Okay, let's get started. So we are going to be finishing Chapter 2 lecture slides today, and then there may be a little bit of time to work on the homework towards the end, but I want to make sure we get enough time to talk about the specific topics for today. And then, let's see, on Monday the plan is that we're going to be going over the first homework questions. So those homework questions are due at the start of class on Monday, and we'll just be basically spending the time talking about them. So the plan will be we'll break into groups of 3 or 4, have you guys sort of discuss all the various questions, and then we'll open it up to the whole class, and I'll basically go around and I'm going to ask you guys to nominate a speaker for each group, and we'll have each group chime in at some point to talk about their answer for one or more of the questions. Okay. So that'll be the plan of attack for Monday. Any questions on anything about that or anything else related to class right now? All right, awesome. So I think this is where we ended last time. That's kind of what I'm remembering. Usually write it down and I didn't. So we were getting to the end of the synaptic terminal, or the end of the axon. And we hit the synaptic terminal, the synaptic knob, the axon terminal. I use all those different terms more or less absolutely interchangeably. So this is where we've got a connection between what's called the presynaptic cell and the postsynaptic cell. So the synapse is this connection between two neighboring neurons that's shown here. And then therefore, if that is the synapse, if the connection is the synapse, then the presynaptic cell is the cell that's sending a signal to that synapse. And then the postsynaptic cell, so this is sort of saying postsynaptic dendrite, or it could be the postsynaptic membrane. This is the cell that's receiving information from the presynaptic cell. These are not just sort of like, dead-end extensions of cytoplasm. They actually have quite a bit of content in them. So they actually have a lot of mitochondria, for example. And therefore, because they've got lots and lots of mitochondria that's present out here, that kind of immediately tells you something about what's going on in these, and that the synaptic terminals have to be actually pretty metabolically active, therefore. They need a lot of ATP, and that's why they've got mitochondria right there generating ATP for them. A big chunk of the ATP demand of these particular structures is going to be revolving around the fact that they're also loaded with these little synaptic vesicles. And the synaptic vesicles contain neurotransmitters. So what will typically happen is you'll have an action potential that'll travel all the way down the axon. And then once it reaches the axon over here, it'll cause an electrical change in the synaptic terminal, which will cause these vesicles to fuse with the presynaptic membrane and dump, which is what's happening right there, dump their neurotransmitter contents into the synapse, where those neurotransmitters can then diffuse over and bind to receptors within the synaptic cleft on the postsynaptic membrane. And that's the typical thing that we'll see happening when we actually walk through and look at what's going on in the synapse. There's also a lot of really unique proteins that are found along the membrane surface of both the presynaptic cell as well as the postsynaptic cell. Postsynaptic cell, largely we're talking about receptors. But the presynaptic cell has a number of interesting proteins that are collectively called snare proteins that are responsible for grabbing hold of those vesicles and delivering them to the surface of the presynaptic cell so they can dump their contents out. I think I mentioned this a couple times. Another thing that often happens near the terminal is you have these branchings, but there's the actual term. So the axons branch to form a terminal arbor. So it's like a terminal tree at the end. Now there's a cool thing, I think, or a big problem if you are a neuron, and that neurons are these ridiculously structured cells. So they are, there's sort of an analogy, if you will, that is, I think this is from your textbook. I can't remember anymore. I think it is. If you sort of assume or scale up the size of the cell body of a neuron to about the size of a tennis ball, then that tennis ball for a typical neuron might have dendrites that would fill up the entire 30-seat classroom. This is about a 30-seat classroom. So you can sort of imagine a tennis ball with dendrites that would be extending out to fill up the entire classroom. That's how large those dendrite populations are. And then the axon would be roughly the size of a garden hose at that point, and then it would extend about a mile in length. So, we're scaling up, and scaling doesn't always work entirely, but this sort of gives you a feel. We always draw these neurons as having these pretty substantial cell bodies. relative to the size of the axon and relative to the size of the dendrites. But really, these illustrations are almost always grossly over-representing the size of the cell body and under-representing the size of the dendrites and the axon. So as a result, you've got this little cell. Well, it's a big cell, but it's still, cells are little. So that's manufacturing almost all of its important materials up in the cell body, and somehow you've got to then deliver that content down to the axon terminals, and as well as out to the ends of the dendrites and such as you're moving proteins and such. Proteins don't last forever. You're constantly having to manufacture new ones, and therefore you need some kind of delivery mechanism to move materials from the nucleus or the vicinity around the nucleus where you have ribosomes and endoplasmic reticulum where you're manufacturing proteins. You need to get those proteins to the end of these incredibly long processes. So the process by which that occurs is reliant on those microtubules. We talked about microtubules in the last lecture. They are basically serving as these little superhighways for walking proteins. And here's a great little GIF animation of a little walking protein. This is a pretty good representation of what we think they look like. They've kind of got a stalk-like domain that ends in a domain of the protein that binds and interacts with a vesicle. And then they have these sort of two globular subdomains that just kind of work like feet. And each step is catalyzed by ATP being consumed. But they seem to do a fairly consistent sort of a walking pattern like this, where they'll lose affinity for the microtubule, undergo a conformational shift, and then they'll gain affinity for that microtubule, and then basically take a step forward. And that's happening in sequence, both one after the other, such that the thing literally just walks down these microtubules. I think they're just adorable. So, so all of these things, you were manufacturing proteins and stuff up in the cell body with all of our typical machinery: nucleus, ribosomes, endoplasmic reticulum, Golgi apparatus. And then what would happen is that if you needed to send a vesicle full of proteins down to the synaptic terminal, to the synaptic knob at the end of the axon, you would package it in a vesicle, and the vesicle would have to get hooked up with a specific kind of motor protein that's called kinesin. And kinesin molecules walk just like this from the nucleus side of the cell down towards the end of the cell, or at least basically they walk away from the nucleus. Technically they walk away from the centrioles out towards the periphery of the cell, including all the way down the axon. Similarly, if for some reason you needed to uptake materials, and this does happen, if you uptake materials from the axon terminal, and then need to send that all the way back up to the nucleus, you have to use a slightly different kind of walking protein. It's called dynein. And then dynein will basically walk the other direction up these microtubules. And this is constantly happening all the time, over and over and over again. In fact, a little video. Hopefully it won't break the recording software, but I think this will be all right. Where are you? Oh, no. Oh, I'm in the wrong chapter, that's why. Sorry. That's just a brain fart. I'm staring here and I can't find my video. Okay. So this little video is showing, it's called mitochondria because it actually shows mitochondria, which also can be pulled up and down these little motor highways of microtubules and motor proteins. And I'm sorry about the recording software in the front. But what this video is showing is you've got basically these little vesicles. So these little spheres are vesicles. This is basically it's a cell, it's a prep where the axon kind of gets flattened onto a single plane on your slide preparation here. And as a result, all of this stuff can be in focus all at the same time using a nice phase contrast microscope. And so these little dots that are moving up and down the axon, those are vesicles that are being carried or pulled by little motor proteins, either down towards the axon terminal or back up towards the nucleus. These longer things that are moving up and down, these are mitochondria. That's a great video. I mean, you can actually see these things moving around within that cell. And some of that this topic comes back up on the homework because this actually turns out to be fundamental to at least one of the things that happens during Alzheimer's disease, where these microtubules seem to be interfered with by the presence of a certain kind of a protein called amyloid protein. So that'll come back up in the homework that we talk about on Monday. Any questions on motor proteins and that kind of thing? All right, so that's some discussion of axons. Dendrites, I think, are also really interesting. We're going to postpone most of our discussion on dendrites until we get to chapter 5, which is the synapses chapter. But just to give you a bit of a preview, dendrites are, I think they're usually underrepresented in any kind of a neuroscience course, and we're probably not going to be any better than that. But you can think of dendrites as this large collection of antennae. So if there's a cell body of a neuron, this is a really nice, one of those little Golgi stains. You can see that if that's the cell body, this is the array of dendrites that's coming in and surrounding that particular cell body and bringing information in. So you can, again, think of them as an antennal array where they're collecting information from the surroundings, and then they're conveying that information electrically to the cell body, which will then ultimately make a decision about is the input sufficient to get me to threshold to fire an action potential in my axon and send signals down to the next cell in line. The membranes of all the dendrites will contain, at least at the synapses, will contain lots of receptors. Those receptors will be specific for the neurotransmitter that's released, or sometimes multiple neurotransmitters that are released at that particular synapse. And there's other cool things you see when you look at dendrites. So for example, these dendrites on this particular neuron, this is a spiny neuron, in that if you look closely at the dendrites, you can see they actually have little, what look like spines. So here's a zoomed in part. So here's, I think that's actually from this part right here. So that's the dendrite right along here. That little breakage there, that's an artifact, something that happened during the slide preparation. That wouldn't be like that in life. But you can see there's lots of these little spines sticking off the edges of the dendrites. Similarly, there's another view of it down here off that particular branching. The dendritic spines seem to be involved with synaptic connections. So you tend to have more dendritic spines on dendrites that have a lot of synaptic connections going to them, and they tend to be localized in the general vicinity. They're not necessarily involved in the transmission of the neurotransmitter from one cell to the next. But one thing they would allow, and there's a lot of open question about what exactly they do, one thing they would allow is for isolation of chemical reactions that could occur in vicinities or right around where neurotransmitters are being exchanged. So you can sort of imagine this is basically a little offshoot of cytoplasm over here that's contained, and therefore you could have specific reactions occurring in there in response to the neurotransmitters that are being received. And that could cause elaboration or changes in the synapses or the function of the synapses at those localized sites. It's also been observed that when there is spine loss or malformation of spines on neurons that are normally spiny, and if you don't have normally shaped or inadequate numbers of spines, those are sometimes associated with mental impairments. So there seems to they clearly have some kind of an interesting function. It may vary from neuron to neuron, but they have some sort of interesting function in terms of the performance of how neurons are able to interact with each other via these dendrites. So I already said this a little bit. So again, some protein synthesis or other kinds of reactions can occur in these dendritic spines. And so the thoughts are, again, it's responding to the stimulation that dendrite is getting. One function of this, I said you can modify synapses potentially or modify the functions of synapses. And this really ultimately is getting at how memory must work, in that memory seems to be literally physical traces of connections or reinforcements of traces reinforcement of connections between neurons has to be present in order for memory to exist. The memory is, we really think of memory as kind of like a physical thing, especially long-term memory, where you've got these reinforced connections between synapses, between neurons, such that when you activate a circuit of neurons, they all kind of get excited at once, and that kind of creates this memory where you're suddenly then aware and able to remember a specific thing, like an incident that happened around a campfire at one point in your life, or the answer to question number 2 on the exam. Okay. Last little bit of the chapter is on classification of neurons. And then from there, we will be ready to start working on homework. So there's many different ways. This is kind of preliminary or setting up what you'll see happen in the rest of the chapter or rest of the semester, in that there are many different ways to classify neurons, and your book will jump between all these different ways to describe different kinds of neurons that we'll be encountering all semester long. So for example, you can classify and define neurons based on the number of neurites that it has. The classic neuron tends to look like this. So we have a cell body with lots of dendrites sticking off of it, and then one axon coming off of it. So we refer to this thing as a multipolar neuron. Multipolar neurons have many neurites coming in contact with the cell body. So there's many different dendrites, and there's an axon in this particular case. They wouldn't even necessarily have to have an axon. As long as they've got more than 2 neurites, more than 2 dendrites coming in, it would be considered to be a multipolar neuron. A lot of neurons, the reason we usually draw neurons this way is, this is kind of the classic view of them. Most of our interneurons look like this. So we'll talk about what an interneuron is in a little bit. Most of our motor neurons also end up looking like this. These are cells that can integrate lots of inputs coming into them up here before making a decision about sending a signal on to the next cell down the line. Another kind of neuron is called a bipolar neuron, and this is where you've got 2 projections coming off of the soma. So here you've got a axon and a dendrite, and that's it. There's 2, so we call it a bipolar cell. The thing about where you typically see these are cells that are functioning within sort of a very consistent shot of circuits. So for example, in the retina, we do see bipolar cells. In fact, there's a whole layer of the retina, if you're learning the names of the cells. We just call them flat out bipolar cells. We don't even try to disguise it or come up with some additional name. We just call them bipolar cells, where they collect input from photoreceptors and then convey that information up their dendrite, down their axon, to the next cells down the line, which in the case of the retina are called ganglionic cells. We also see these things in the olfactory epithelium. We've got bipolar cells, and so they're often involved in some kind of a sensory pathway. Then we also have unipolar cells. Unipolar cells is where there's just one neurite sticking off. There's one process coming off of the cell body, and these guys are a little weird, where this is a classic example of one of these somatosensory neurons, where they're picking up touch or temperature or pain information from elsewhere, or from somewhere in the skin, for example, and then sending that information up the axon out to the targeted destination. And the thing about it is the cell body is just kind of marginalized or pushed off to the side in these cases. So here is a cell body. It does have one connection to it, but for the most part, the signal will just sort of bypass the cell body as it's being conducted up this axon. Most of our general somatosensory neurons that we'll be focusing on when we get to the somatosensory system are actually these unipolar neurons, and they can be really long. You've got neurons that are in your fingertip that will send signals all the way up into your spinal cord and up to the medulla of your brain. And it's all one big, long, unipolar neuron. Yeah?
Is there an advantage of it not passing through the cell body, or is that?
I don't know. It does have an effect in that this creates these peripheral ganglia. So out in the outside of your central nervous system, there are little collections of cell bodies that usually they're right outside the spinal cord. One thing it does allow, I guess, is it does allow for a continuous signal to be sent from the receptive endings up here, and it's just all one series of action potentials that goes all the way up to the destination. But yeah, why couldn't you put the cell body right there and do the same thing? I don't know. I haven't really seen anything on that. There's another hand somewhere. I could be seeing things. That's all right. Good question. I don't know the answer. Okay, there are many other ways to classify neurons, though. So even A&P textbooks do talk about classifying neurons based on number of neurites, and so we just went through that. But you can classify cells based on the layout of their dendrites. So when we start talking about the brain, in particular when we start talking about like the primary motor cortex or some of the other areas, we'll run into stellate cells and pyramidal cells. Stellate cells are just, stellate refers to stars, and it basically, in this case, what you have are dendrites that are right about in the appearance of something like a star. It's just sort of like a big burst coming out of the central cell body, which would be right there, if I can hold my pointer still enough. Whereas pyramidal cells, or pyramidal, I can't say it that way. I always say pyramidal. That's how I was raised, but other people say it otherwise. There's the cell body, and they'll have this series of dendrites that'll extend on upwards. But the thing about it is that if you look at the sort of dendrites as a whole, it kind of forms a big triangle here. And so in this case, we refer to this thing as a pyramidal cell, and that it looks, kind of like a big triangle or a big pyramid. It's a three-dimensional structure, so it looks kind of like a big pyramid heading up. These things have different functions. So these guys are often going to be, sometimes they don't even have axons. They're integrating information from many different sources if you're a stellate cell, but they're then sending that information to localized areas. So they're not going to be communicating very far. They're going to be communicating with other cells that are around them in their vicinity. Whereas these big pyramidal cells, they're integrating information from many different sources, sometimes across multiple layers of cerebral cortex. They're integrating that information at the cell body, and then these are the cells that would have really long axons that would extend all the way, potentially even all the way down your spinal cord. So when we look at the primary motor cortex, we'll see neurons that look like this, that'll be integrating information up there, and then have an axon that sends signals as it integrates that information down your spinal cord towards the motor neurons that are found where the cell bodies are found in the spinal cord, that then go off to your specific muscle cells. So this is a common kind of cell we'll see there. So again, you're basically just naming, though, in this case, you're naming the cells based on the shape of the layout of their dendrites. You can classify neurons based on whether or not they have spines. So you can have spiny versus aspinous dendrites, as we just saw in the prior discussion of spiny dendrites. And you can also classify them by function. This is a very common thing to do, and you've probably seen these terms before as well. So you can classify based on what is the function of this neuron to your nervous system. And through this classification, basically you have three different varieties. You have sensory neurons, which are sometimes also called primary neurons, or primary sensory neurons. You have interneurons, and then you have motor neurons. Sometimes we specifically would call these lower motor neurons. And the key difference is that the sensory neurons are picking up information not from other neurons, but from the world around them. So they're picking up, in this case, they're kind of showing them as, retinal cells, or they could be tactile cells, where they're picking up information from either the external or internal environment, but not from other neurons, and then they're sending that information to other neurons. Interneurons would be completely nestled between other neurons. So they're getting information from neurons and then sending it to other neurons. So interneurons are between sets of neurons, hence the term inter. And then finally, we've got motor neurons, which are getting information from other neurons and then carrying it to some kind of an effector. So this could be a skeletal muscle, it could be a gland, it could be the heart, it could be whatever it's destined to be interacting with. But it would not be a different, another set of neurons in this case. Other things, you can classify by axon length. So you can classify, there's this general classification that goes back to that guy Golgi we talked about that developed the Golgi stain, where you have Golgi type 1 neurons, which are sometimes referred to as projection neurons. These tend to be really long axons. So there's that pyramidal cell that I showed you a little bit ago that's collecting information and then sending really long axons that go all the way down your spinal cord. That would be an example of a Golgi type 1 cell because it has a really long axon. These are typically communicating at least across the brain, if not from the brain to somewhere else. So they're going over long distances. Whereas Golgi type 2 cells would be what are referred to as local circuit neurons, where they're just sort of communicating within a small network that's all closely in close proximity to one another within the brain. And then finally, you can always classify neurons also by the neurotransmitter that they release. So different kinds of neurotransmitters have different neurons that are that they're named for. So for example, if you're a neuron that releases acetylcholine, which includes pretty much all the motor neurons, it includes a lot of our parasympathetic nervous system neurons, it includes some of the sympathetic nervous system neurons. Acetylcholine is a really important neurotransmitter, and you've probably heard about it before. All those neurons that release acetylcholine are called cholinergic neurons. In contrast, if they release norepinephrine, which is another really common, really important neurotransmitter, they're called adrenergic neurons. And there's other names for all the other kinds of neurotransmitters, glutaminergic neurons that are releasing glutamate, and so on. any questions about any classifications of neurons or anything else from this chapter? Good. So at this point, then, we're done with the slides. So we've got 25 minutes or so, I think. 24 minutes. So, I'm not going to force you to, but you are welcome to pull out the questions, start working through them. If you don't have your lecture textbook with you, if you at least have a phone or your computer with you, you can watch the Inner Life of the Cell video and start working through that worksheet. There's a lot you could do. But this is time basically for you guys to be working together, or if you want to get out because it's a beautiful looking Friday afternoon, you can do that too. But I will be here if people are going to stay around.